Okay, so now I'm going to demonstrate how you would have processed your bugs, so your macroinvertebrates that you got from the stream. So remember, we sampled the stream, 10-metre stretch of the bank using the net, and then we've did a 20-minute live pick where we've collected anything that was moving or obvious. And then we put it in our jar with ethanol, and I'm not telling you the answer for the ethanol question, but what we got is our sample. So now we need to identify what we found. And so we're here in the lab, and I've just got a dissecting microscope. This allows us to see a bit more detail, and I'm just pouring the sample in. Hopefully it doesn't flood. Try and get all that out into our jar. So even though we've done a live pick and everything in here should be a specimen, an organic specimen, sometimes you don't know, you can't see as well, and so we need to confirm that everything we have. So here's our sample. This is from Yosemite Creek, and you can see maybe the sample there. And so what we then need to do is kind of identify what groups of organisms they belong to and how many. So while I mentioned when we're in the field that we're not after an exact number, it's relative numbers that's important, because that helps us in understanding the relative abundances of these organisms. So I've got, as I said, a light microscope here, and I've just checking under there, and I see lots of little critters. So what are they, and how are we going to identify them is the question. And so we've got this guide, and this is on iLearn, and so you have this exact document on iLearn. It tells you about the surveying, and what you'll end up doing is having to create a signal score. So signal is a measure for the quality or the health rating of your macroinvertebrate community at each site. And so what we'll talk about in class is how to derive those numbers, but in essence what you're going... Can we pause? Sorry about that. That was an alarm going off in the lab. No need to panic though. It wasn't a fire alarm. It was the fume hood telling us there was some issues. So anyway, we've dealt with that. So back to sorting bugs. And so yeah, you have this guide online. At the back of this guide, so it tells you how to create your signal score, but more importantly, and this is what we're going to be doing now, is there's a whole photo guide, and you can go to, this is part of the Murray-Darling Freshwater Research Centre has created this guide, and so you can actually go to their website and have a look at this as well if you want more detail. And so basically what it's done is created a measure for each of the major taxonomic groups that you might get in a stream, and given them some code. And so you'll see, with the photo here, that there's some little bars down the side that tells us about their habitats, or on either side on their habitat, their trophic level, whether they're a herbivore or a detritivore, things like that, their functional group, what role they play in the community, and where they live. So, are they in flowing environments or pool environments? And then it gives us a signal score for each one, more importantly. So a number between 1 and 10. And so it tells us immediately, if you see something in that group, how sensitive or not it is to disturbance or stresses in that system. There's the alarm again. I don't know. And so what you'll end up doing is calculating like the signal scores for each individual family you get. So there's photos, and you've got to remember that the photos are only representative of one individual or one particular species in that family. And so sometimes we get things that look kind of the same but aren't exactly what is in the image. So we kind of look for key features that are common amongst that, and that is an indication that it belongs to that family. And so you'll have to assign what family these bugs belong to. So we will send you some photos of some of these critters, and you will have to use this guide or other sources to determine what family it is. So it's really the family level that gives us enough taxonomic differentiation about the condition of that stream. So if we just dealt at the class or the order level, it wouldn't give us enough information. So we can go down the family. We don't have to go all the way down to species to do that. That would be very onerous, and you need a taxonomic degree to do that. And I'm not skilled in that regard either, but it's highly specialised skill. So the family level is sufficient to give us enough idea of the health of the streams. So basically what I'll be doing now is pulling out the different groups, and we'll show you some photos in a minute. And there's key features that you'll be able to see. And so so if you remember if you've seen the field videos, we had little critters living in stick casings. So they're called caddis fly larvae, and so they're in the Trichoptera family, okay, or Trichoptera order, of which there's many different families. And so you would need to decide which Trichopteran it belongs to, which, as I was saying previously, the Trichopterans really differentiate by how they make their casings. So some are made out of rocks, some are made out of sticks, some are made out of leaves, and so we can see quite easily what family they belong to. But I'm going to leave that for you to do. So all I'm going to be doing is picking out the groups and recording how many there is in each of those taxonomic groups, and then we'll send you the photos and you have to identify what family they belong to. And so remember, you're going to have to record the class and family, but we'll give you a bit more detail of that in coming lectures. Okay, so we've got here an example of one of the bugs, and I wanted to point out some key features so that, things to look for. Now, the obvious things are its legs. So remember, all insects are hexapods. So that means they've got 6 legs, okay? So we can see, obviously, 3 pairs, and that is, tells us, one, it's in the Insecta, class Insecta. So other things we can see around the head here, we can see some large kind of bulbous eyes. You may be able to pick up in the image, there's some very short antennae there. So that some insects have very long antennae, others are short. So this species and this group, this family is known to have very short antennae. So it's very in between kind of the nostril area. If we turned it over, we'd see there's kind of actual biting or chewing mouth parts, and so it tells us how this animal feeds. The other thing that's distinctive is the long, kind of elongate body, so the abdomen region, and it comes to the tail area, and it has two little pincers, if you like. They're not pincers as such, a two-pointed tail, very short. And so, you know, we're looking at the legs, the head shape, the general body shape, and the tail are key features of most of our bugs. Okay, so head, tail, and general body shape are really the distinguishing features. So if I now move that out to our, swap that out to our guide, and I'm going to switch it out to see how this goes. Yep. We can see that, and because I've already looked, you know, by flicking through the guide, you can see this one here is very similar in the body shape, the head shape, short antennae, short tail region, obviously 6 legs. So this is, you know, the key features are very similar, and although the colour's different, once again, colour is probably not a good indication for most things. It's the body shape and the key features is what you need to be looking at. So this tells us that it's in the Odonata, which is the dragonfly larvae, and the family here is Acinettidae. So it has the order and then the family down the bottom here, and then the signal score is a 4 for this one. So you would note down those, anything along the bottom there, and if you want to put the common name, you can put that as well. Okay, so that's just one example. I'll pull out another critter just to give you another example as well in a minute. Okay, so I've put another critter that I picked out of the microscope, and this is one of those ones we saw crawling around that were encased in this like stick-like casing or cocoon. And what we can see in the image here is it's pretty much its entire body is covered with this stick casing, but its head and its legs are sticking out. And so you don't get to see much, you only get to see really the head features and those limbs. But that is enough to tell us a bit about where this species might come from. So once again, we look at the feature of the casing in this case, the limbs, and so we note that the last pair of limbs are actually quite long and protruding well above the head and much longer than the other limbs in this particular individual. So we note these features, and then we're going to go to our guide. So I'm going to move that out of the way. So I've looked through the photo guide, and I've come across animals that are in casings, and you can see 2 examples here. So firstly, the top one has lots of little what look like rocks, and they are little rocks made out of casing. This one here is out of sticks, so it tells us immediately, you know, that maybe this is our guy that we're interested in. And there's other images in the photo guide, but these are the two that look similar in the length and shape. But if you remember back to the actual critter, this bottom one, you can see, hopefully in the image here, you can see these limbs protruding well above the head. This is the third pair of limbs protruding well above the head, much more than the one above is doing. So if the casing's not giving it away, then the body and head, sorry, limb shape is also, you know, a key feature. So to me, I think our specimen is this one. So this is a Trichopteran, which is a caddisfly larvae. So and the family is Leptoceridae. So they're the key things. It's a 6 on the signal score. So the higher the signal score, the more sensitive the animal is. So if it's a polluted environment, you don't get a lot of, you know, 6, 7, 8, 9 type species appearing. So we'll talk a bit about that in another video. But the common name, longhorn caddis, it pretty much comes from the fact that its limbs look a bit like horns protruding from the casing. So even though they're actually legs, not horns. So anyway, another example of what to look for when we're identifying our bugs.